nucleic acids are the building blocks of the DNA and RNA. So that means this whole structure of the DNA is made up of these chemicals, these compounds, these structures, these molecules, which are known as the nucleic acids. Okay, and here you can see there are some letters written in between both the strands or the helices of the DNA. So these are the actually uh, various types of different types of the nucleic acids. How these nucleic acids are further classified and what these nucleic acids are actually. So these nucleic acids are molecules that store information. So that means whatever do we carry in our body, whatever the information that we transmit to the offsprings is carried in these nucleic acids. And these nucleic acids are written in the transcript of some alphabets, right? As these are known as A, T, C, G, T, U. So if you are aware of the computer language or the coding language, computer language is actually encrypted. Computer doesn't read this text in this form. ठीक है, कंप्यूटर के लिए ये कुछ और इंफॉर्मेशन है, ये encrypted फॉर्म में होगा. So similarly, our DNA is made up of these letters information arranged in different sequence of orders. Now, there are two very famous types of nucleic acids that we have in our body. Number one, it is the deoxyribonucleic acid, which is known as DNA, and a ribonucleic acid. So, later half of the chemical is the same as that of the nucleic acid, but one is the ribo and one is the deoxyribo. What is the difference in between them? The ribonucleic acid, we know that the ribose is a type of a pentose sugar. It's a carbohydrate. So when these nucleic acids get attached or combined with the ribose sugars, they make ribonucleic acids. And it is in the oxygenated or oxidative form. But when the same ribonucleic acid is actually deprived of oxygen at a particular location, it becomes DNA or a deoxy form of the nucleic acids. So this is what I'm talking about actually. This is a pentose sugar, which is ribose. And at this particular position, so we do not see any oxygen over here, so certainly it can be a part of the DNA. Okay. Now, the basic ingredient or the recipe of the nucleic acids lies within the nucleotide. And the nucleotide are what? The nucleotides are the structure which do contain a pentose sugar, a nitrogenous base, we will talk about it in a while, and a phosphate group attached with it. So when a pentose sugar and a ribose base and a nitrogenous base is connected with a phosphate group, it becomes a nucleotide. Okay, we call it nucleotide. So when phosphate is there, it is a nucleotide. And when it is devoid of this phosphate, when there is no phosphate group attached with that, we call it a nucleoside. So this is the difference in between the nucleoside and the nucleotide. Okay. Now, what about these 1, 2, 3, 4, 5? Can anybody tell me what are these numbers and what is their importance? These are the carbons. These are the carbons. Yes, these are the carbons present within the pentose sugar. But if you notice that these carbons are written differently in this sugar, you see a small dash or apostrophe S-like structure. What these are known as? How do we call them? How do we pronounce them? Yes, these are the primes. So every carbon is designated as prime number. So we read it or we call it 1 prime to 2 prime to 3 prime to 4 prime to 5 prime. And it is important to know that what actually primes are, because the whole process of DNA replication and then transcription is based actually on these uh, prime numbers. Okay, so this is the basic chemistry of the nucleic acids. Now let's talk about the nitrogenous bases. We discussed that we have a sugar which is pentose, which we have a phosphate group, and let's talk about this part of a nucleotide, which is a nitrogenous base. What are these nitrogenous bases? We have two types of nitrogenous bases in general in a molecule of DNA. They can be either the purine bases, purine nitrogenous bases. The purine type of nitrogenous bases are the adenine and guanine. So adenine and guanine are the purine bases which have two ring structures connected together. Okay, and these purines and these purine bases, which are adenine and guanine, they are found throughout DNA or RNA molecules. So they are there within the structure of DNA and RNA. I would like you to notice the location of this amine group and oxygen in these structures. We will discuss their importance somewhere in process of transcription or maybe in the repair defects. Okay, so these are the structures of purines which are present in DNA and RNA. The other set of nitrogenous bases that we have are the pyrimidines. We have three types of pyrimidine bases, which are the cytosine, thymine, and uracil. And these pyrimidines are actually single ring structure, and also notice the position of NH2 over here, right? And oxygen over here. But one thing is to remember that in case of pyrimidines, that this cytosine is present in both DNA and RNA. Cytosine is present in both. Thymine is present in DNA only, and uracil is not present in DNA. Uracil is a type of nitrogenous base which is not a part of the DNA. Uracil is part of the RNA molecule. So by having uracil in the RNA, in the structure of RNA, or in a structure of a polynucleotide chain, you can identify that this polynucleotide chain belongs to the DNA or the RNA, because in the DNA there will be no uracil. Okay, so these are the two types of the nitrogenous bases that we have in the DNA. Okay, now there is some terminology that we often use to describe or explain the nitrogenous bases and the structures present within the DNA molecule. We talked about the nitrogenous bases, which are the adenine, guanine, cytosine, thymine, and we talked about the nucleosides and the nucleotides. What we have discussed, that nucleoside is a type of nucleic acid in which we do not have any phosphate group. But what do we have in the nucleoside? We only have a nitrogenous base and a pentose sugar. So if the structure is of RNA and the type of base is present is adenine, and this adenine is attached with a pentose sugar but no phosphate, we will call it adenosine. Adenosine. And if this same adenine is get attached with the pentose sugar ribose and a phosphate group with it, so we will call it adenosine 5 prime monophosphate. Now what does this 5 prime mean? This means that in that structure this phosphate group is attached to the carbon number 5 of the adenine base. Okay, so this is the difference. So same is the case with the guanine. It will make guanosine or guanosine monophosphate. Cytosine will make cytidine or cytidine monophosphate, or uracil will make uridine or uridine monophosphate. So we go similarly with the DNA: deoxyadenosine, deoxyguanosine, deoxycytidine, and deoxythymidine. So these are few terminologies that we use in order to study the structures of DNA. Okay. Now, we talked about that DNA is a double helical helical double-stranded structure. So that means it has two strands. Okay. Now, how to read that primary structure? How to read that single strand of the DNA molecule? We start reading, or we, by convention or by design, we start reading the polymer of DNA in the direction of, as it has a free 5 prime phosphate group, as you can see over here, that this is the ribose sugar, this is the base adenine attached with this ribose sugar, and at the carbon number 5 of this pentose sugar, a phosphate is attached. Okay, so what we do, we start reading from this end, which is a 5 prime end. So free 5 prime phosphate group at one end and free 3 prime hydroxyl group at the other end. As you can see, this is the free 3 prime hydroxyl group. Okay, so we start reading from 5 prime to 3 prime direction of the DNA strand. So this is how by convention we start reading the DNA template. So if this is a DNA, or this is a fragment of a DNA, or this is the DNA template, the sequence is read from the free 5 prime end using the letters of the bases. Okay, so we will read it like 5 prime A, C, G, T, 3 prime. So this example will make a transcript like that. So this is 5 prime, this end, 5 prime A, C, G, and T. So by convention or by design in molecular biology or genetics, we read the structure of the DNA or RNA templates in this way. So 5 prime to 3 prime and the number of bases in between specify us a direction of the template actually. Okay, let's take an example of the structure of the DNA. May I ask you, okay, which structure is this? Is it DNA or RNA? Is this DNA or RNA? It is DNA. How can you say that it's DNA? Why not? If I say that it's RNA, it has thymine, okay? If it would be a structure of RNA, then what would it have? It uracil, okay. So if it would be a RNA structure, instead of thymine, we would have the uracil in place of this. So what we are looking at this, the same nitrogenous bases, the same purine, which is adenine type, the same pentose sugar, and the same phosphate group. So that means this is the one nucleotide. This one is the second nucleotide. This structure is the third nucleotide, and this structure is the fourth nucleotide. So this is how, when the nucleotides are joined together, get connected with each other, they make a polymer. And this is how DNA templates or the DNA strands are built up or synthesized. Okay. So we will read this template in a very similar fashion. That is, we will start reading it from 5 prime A, C, G, T, right? And 3 prime. So this is the direction of the template. But the point that I'm trying to make here, the point that I'm trying to emphasize on, if you look at this, these are the 4 different types of nucleotides. And these nucleotides are connected with the help of a ester linkage. This ester linkage is known as the phosphodiester linkage or the phosphodiester bond. And if you notice the direction of this phosphodiester bond, or the location in between the 2 carbons of the pentose sugar, this phosphate bond is formed, is different than the 5 prime to 3 prime direction. If you can notice that this is the phosphate group, and this phosphate group is attached where? At the 3 position, 3 prime carbon of the sugar, and where the next nucleotide is attached. Next nucleotide is attached on the 5 prime carbon, 5 prime carbon of the pentose sugar. So that means in between 2 nucleotides, the phosphodiester bond is formed in a direction of 3 prime to 5 prime. Got it? Got it? So can anybody repeat the direction of the transcript? Yes, the direction of the transcript. Anybody? Can anybody repeat the direction of the transcript? 5 prime to 3 prime. And the direction of the phosphodiester bond? 3 prime. Good enough. Good enough. So this is the point. This is the point. And you need to remember that. We will read the transcript or the template in 5 prime to 3 prime, and we will read the direction of phosphodiester bond from 3 prime to 5, 3 prime to 5 prime. Okay. Now, one of you talked about that it is complementary. Let's look at the concept of complementarity or the anti-parallelism in the DNA structure. I take it as one strand of the DNA or the one template of the DNA, and I assume that this is the 5 prime end of the DNA, 5 prime phosphate, and I assume this is adenine, this is cytosine, this is thymine, this is guanine, and this is the 3 prime end. So DNA molecule is formed by joining of the millions and millions of nucleotides. And these millions and millions of nucleotides are joined together with the help of phosphodiester bond. And one thing to remember that these nucleotides are attached with each other, connected in between the two strands of the DNA in anti-parallel and complementary fashion. So if I assume that this is the 5 prime end of this strand of the DNA and this is the 3 prime end, so what would be the anti-parallel strand? In anti-parallel strand, where will be the 3 prime end? Anyone? Anyone? Yes. So 3 prime end will be this one. If it is if this is 5 prime, this is 3 prime, so anti-parallel will be 3 prime here and 5 prime here. So this is the concept of the anti-parallelism in the DNA. We will see in a detail. The bases always pair up in the same way. These are complementary to each other. So when I say these are complementary, that means adenine will always bond with the thymine. It can never bond with the cytosine. It is complementary for the thymine. Thymine, adenine for the thymine, and cytosine bonds with the guanine. It will never bond with the adenine. So every base has a complementary base pair associated with it. So anti-parallel 5 prime, 3 prime, 3 prime to 5 prime, and complementary adenine with thymine, cytosine with guanine. Okay, look at this. This is one strand and opposite strand you can see. This is 5 prime, this is 5 prime, this is 3 prime, adenine, cytosine. Now, this become 3 prime, thymine in front of adenine and guanine in front of cytosine. And same is the case for rest of the template to be built in this manner. So there cannot be a mismatch. There cannot be a attachment of a base which is not complementary. Now, what about the central dogma of the molecular biology? What that central dogma tell us? This central dogma of molecular biology explains us 3 things. Number 1, the process of replication, transcription, translation, and finally a protein product. Now, what is the process of replication? If you look at the arrow, it is a DNA. This is the DNA molecule. And if you look at the arrow, that DNA is made from the DNA. So that means it's a replica. It's a Xerox of a DNA molecule. So in the process of replication, what cells do, what DNA do, DNA make a copy of itself, make a replica of itself. Why? To make a transcript, to copy this information before the cell division or to transmit this information to the generations. So it has to be replicated first and then transcribed into a language. And that language is translated into a protein. So these 3 processes, the copying of DNA, then DNA is converted into the RNA or RNA transcript, and then that transcript, which is RNA, is actually translated into a product, which is a protein. So we will look at these processes in detail one by one. So today we are talking about the process of replication. So what's happening here actually? The process of replication is actually the copying or xeroxing the information, the exact information which is present in these strands of the DNA, in these nucleic acids, which are further transcribed into RNA, and RNA, after its processing, makes some new proteins which are needed for the normal functions of the body. Now, this is the structure of a chromosome under normal conditions, and to your surprise that this is the single stranded structure. Before that, maybe you are aware of a double structure or X-like structure of chromosome, but chromosomes are these single stranded type of structure. Do you know that how these single stranded chromosomes are become or converted into a double stranded structures? Do you know that? How they become double stranded? Yes, they become double stranded by the process of replication. The process of replication make them double stranded. How? If you look at this single stranded chromosome, they uh, they prepare themselves to divide the DNA. The DNA is copied and it replicates itself into the two strands, these two strands. The ends of these strands are known as the chromatids. And this has to have taken place during the cell cycle, before the cell division, every single segment or fragment of the DNA need to be copied so that the complete set of information from the parents or the ancestors can be transmitted as a hereditary character to the offsprings or to the new cells being synthesized in the body. So therefore, prior to cell division, it is must that DNA need to be replicated. Here you see that the cell division takes place in the M phase of the cell cycle where mitosis takes place, and where the cell divides actually. So it is important that the process of replication has to be undertaken, has to be completed in which phase of the cell cycle? In S phase of the cell cycle. S phase is before the S phase is before the M phase of the cell cycle. So when the cell copies and preserves all of its information, only then it is allowed to be divided so that the same information which was present in this DNA, which was replicated, making copy, can be transmitted to the daughter cells as well. So this is the process by which uh, by which the cell divide themselves. Now, what about the replication itself? It is the process of producing two identical replicas from one DNA molecule. So, why two identical uh, um, um, replicas from each single strand of the DNA, right? So, every single strand has to be copied, has to be replicated. If you can see this, these are the two strands of the DNA. And if you notice this yellow one, this yellow one is actually a new strand or the daughter strand which is being copied from the parent strand. Okay, so it has to be copied for the purpose of biological inheritance. The information which is copied is transferred. The DNA replication is a semi-conservative type of replication. Why do we call it semi-conservative? We call them semi-conservative because it conserves exact information. This is the parental DNA, the two strands of the DNA, and these are the replication cycle in which the first strand is copied, as you can see over here, and in the second half, the second strand of this one, this was, it is also copied. So it is semi-conservative type of the replication in which all the information is conserved or copied exactly in the same manner. Okay. Now, what happens? in the DNA structure itself. Before replication to start, there is a location or site of origin, or the site of replication is identified within the DNA structure, within a molecule of DNA. And this V-shaped location or the V-shaped site of replication is formed when the two strands are separated. So what we are talking, that they are the two helical interwoven strands. So to make a copy or a replica, both the strands need to be separated. Both the strands need to be unwind. And when they unwind or separate, there is a region formed which is known as a V-shaped form or a replication fork. And this is the place where the active synthesis or the where active copying of the DNA takes place. If you can look at this direction, in this diagram, you can see the two strands, 5 prime to 3 prime, okay, and 5 prime to 3 prime, anti-parallel to each other, right? Both the strands are anti-parallel, okay. Now, what will be the direction of replication? In which direction replication will start? Three prime to 5 prime. Why in 3 prime to 5 prime? No, the direction of replication is always 5 prime to 3 prime, okay. It is the only direction of the phosphodiester bond, which is the 3 prime to 5 prime. Now, I explain you. If these are the two strands anti-parallel to each other, and this is the site of replication or origin of replication, and what we call that, it makes a replication fork. If you look at this structure, this diagram, it looks like a fork. That is why we call it a replication fork, or you can call it replication bubble as well. It has a site of replication which is moving towards the replication fork, right? Towards the replication, if you see this. So if it is moving, if it is the 3 prime to 5 prime strand, and the new daughter strand has to be synthesized in the direction of the replication fork, it has to be anti-parallel, okay. And if it has to be anti-parallel, so from where it will start? What will be the direction of replication then? Yes. So it has to be anti-parallel. So it has to be start from in 5 prime to 3 prime direction. So when the direction of replication is 5 prime to 3 prime in the direction of replication fork, we call it that strand which is being synthesized in the direction of replication fork. We call it a leading strand, or the strand which is being synthesized in the direction of away from the replication fork, opposite to the direction of replication fork, we call it a lagging strand, okay. So this is the difference in between the leading and lagging. Leading strand in between, towards the direction of replication fork, synthesis of new DNA, and lagging strand away from the replication fork. There is another difference I will explain in other diagram. Now, let's talk about the enzymes. What enzymes do we need for the DNA replication? We need helicase, DNA helicase, because DNA is which type of structure? Since it's a helical structure. So before replication, what we need to do? We need to unwind this helix. So this helix will be unwind with the help of helicase. So we need the first enzyme, which is DNA helicase. What DNA helicase will do? DNA helicase will separate out the two strands, remove the helical structure, right? And this there binds some single stranded DNA near replication fork and move into neighboring DNA strands, forcing them apart, right? This is how. Imagine or consider this is one strand of the DNA, this one is the other, and this is the helix, both the templates. And this is the enzyme DNA helicase. What this DNA helicase is doing? This DNA helicase is separating both the strands. As you can see, it is removing the bonds between the nitrogenous bases, and the strands is opening up. So it's opening up with the action of DNA helicase. Now it's becoming no more helical. The second important enzyme is the DNA topoisomerase. Topoisomerase, what's the job of the topoisomerase? When the DNA helix uh, unwind or separate out, so what happens? There is a formation of loops or coils adjacent to that. आपने वो टेलीफोन की कभी वो देखे हैं ना कनेक्शन, अगर आप उसको केयरफुली वो ना करें तो उस हेलिक्स, वो भी हेलिकल स्ट्रक्चर होता है, सिंगल हेलिक्स होता है वो, अगर वो ना करें तो उसके बीच में क्या होता है, लूप्स बन जाते हैं, टर्न्स बन जाते हैं, और अगर आप उसको बीच में से कभी इस तरीके से वो पास ऑन करें तो वो लूप्स जो हैं और टाइटर होते चले जाते हैं. तो दैट टेंशन इज रिमूव्ड बाय द डीएनए topoisomerase. So what it do, it removes the supercoils. If you can see, this is the strand of the DNA, and what helicase is doing, helicase was separating them out. Helicase इनको क्या कर रहा था, अलग अलग कर रहा था. तो as a result of this separation, what was happening, these supercoils or superfolds are being formed within the replication fork or within the direction of the replication. So these supercoils are actually removed by the action of topoisomerases. तो job of the topoisomerase is कि वो जो supercoils बनेंगे as a action of helix or unwinding in supercoil remove करेगा DNA topoisomerase. So we talked about two enzymes. Okay. Now we will talk about third enzyme, which is DNA polymerase, which is RNA polymerase, or you can call them primase or the DNA polymerase. Now, look at this. What I told you that we dealt in the two directions of the replication, either towards the replication fork, and towards the replication fork was the direction. वो क्या थी? Leading strand, or away from the replication fork. Okay, so the primer is synthesized, and primer is needed to start the process of replication. Okay, and this red fragment is known as the RNA primer, uh, or the primer. This primer is synthesized by the primase enzyme. Okay, now what you notice here, in the upper strand, which is in the direction of replication fork, जो इस तरफ जा रहे हैं, जो कि लीडिंग स्ट्रैंड में है, यहाँ पे एक प्राइमर लगा, इमेजिन दिस इज द प्राइमर, और वो प्राइमर ने एक कंटिन्यूअस सिंथेसिस विद द हेल्प ऑफ डीएनए पॉलीमरेज 3. डीएनए पॉलीमरेज 3 एक कंटिन्यूअस नया स्ट्रैंड डीएनए का सिंथेसाइज करता चला जा रहा है विद इन द डायरेक्शन ऑफ replication fork. So this is known as continuous synthesis of DNA within the replication fork. Okay. Somebody has raised hand. Raised hand for a question or attendance purpose. Okay. You can raise your hands if you want to mark your attendance. Please raise your hands. Okay. Oh my God. Right. Now, what's happening in the other direction? That is the direction which is away from the replication fork. Okay. What's happening there? This type of synthesis of DNA is discontinuous. It is not continuous. RNA comes and it make a small fragment or portion of the DNA and then discontinue. Then other RNA comes in between, and then some synthesis of new DNA strand, then other RNA primer, then some other. So this type of synthesis of DNA is discontinuous or non-continuous. So this is the second difference of DNA replication in the direction of replication fork, or in the leading strand or in the lagging strand. So I think I stop my class here. Tomorrow we will continue from the same slide. Okay. If you have any questions, you can ask.
वालेकुम सर।
वालेकुम सलाम।
सर ये जो आप कह रहे हैं फोर्थ की साइड पे, मुझे ये वाली बात नहीं समझ आ रही। फोर्थ
क्या
की
जी?
साइड का आप कह रहे हैं कि जो है फोर्थ की साइड पे हो रहे हैं डीएनए फोर्थ की साइड। सर आप एक मिनट स्लाइड पीछे कीजिएगा।
आप इस स्लाइड की बात कर रहे हैं या आप इससे पहले वाली स्लाइड की बात कर रहे हैं?
सर आप ये कह रहे थे कि जो भी कुछ हो रहा है वो रेप्लिकेशन, मतलब आपने दो तरीके की बताई थी। एक जो स्ट्रैंड बन रहा था वो रेप्लिकेशन फोर्क की तरफ बन रहा था।
स्लाइड की बात कर रहे हैं।
जी सर। सर
ओके।
आप कहते कि एक एक जो वो बन रहा है वो रेप्लिकेशन फोर्क की तरफ बन रहा है, तो मतलब रेप्लिकेशन फोर्क की तरफ बात का क्या मतलब है कि
इसका मतलब ये है कि ये एक रेप्लिकेशन फोक बन गया ना, दो
जी
स्ट्रैंड्स
सर।
हो गए ना, ठीक है।
जी
ये एक डीएनए मॉलिक्यूल था, इसके दो स्ट्रैंड्स थे। अब हर स्ट्रैंड की रेप्लिकेशन
जी
होनी है
जी
ना,
जी सर।
ये ये इस वाले की भी कॉपी होनी है और बॉटम वाले स्ट्रैंड की भी होनी है, ठीक है। अच्छा, ये
जी
जो
सर।
होती है बाई डायरेक्शनल होती है, ऑपोजिट डायरेक्शन में होती है, ठीक है। तो अब अगर रेप्लिकेशन फोक बन गया और रेप्लिकेशन ऑपोजिट डायरेक्शन में होगी, बाई डायरेक्शनल होगी, तो क्या होगा? और उसकी डायरेक्शन हमेशा कहाँ होगी? 5 प्राइम टू 3 प्राइम होगी ना। तो इसका अगर 3 प्राइम एंड यहाँ पर है, तो जो नया डीएनए बनेगा और वो एंटी पैरेलल बनेगा
जी
इसके, तो वो कहाँ से शुरू होगा?
ये यहाँ जहाँ सर्कल बना है 5 की तरफ से।
ये यहाँ से शुरू होगा, जो ये आपको ग्रीन ग्रीन प्राइमर नजर आ रहा है, ठीक है।
सर तो ये तो 3 प्राइम एंड ही है ना।
बेटे, ये 3 प्राइम एंड तो इस वाले का है ना, पेरेंट स्ट्रैंड का है ना, इसका
अच्छा
एंटी
सही।
पैरेलल बनेगा ना।
अच्छा
अब
अच्छा अच्छा सही सही।
शुरू में हमने बात ये की है ना कि डीएनए के स्ट्रैंड्स हमेशा एंटी पैरेलल होंगे और हमेशा कॉम्प्लीमेंट्री होंगे। तो अगर आपको एंटी पैरेलल स्ट्रैंड बनाएंगे आप, तो 5 प्राइम से शुरू होगा ना ये।
ठीक है सर।
तो
और
अगर ये
सर
यहाँ,
नीचे,
जी
और
जी बोलो।
नीचे वाला जहाँ दूसरा ग्रीन वो है उधर से शुरू होगा क्योंकि
अब नीचे वाले स्ट्रैंड का 5 प्राइम एंड ये है, तो इसका एंटी पैरेलल जो बनेगा, इसका एंटी पैरेलल 5 प्राइम यहाँ से बनना शुरू होगा। तो यहाँ पे क्या होगा? ये प्राइमर आके लगा, प्राइमर फिर इनके बेस पेयर्स इंसर्ट करता जाएगा। मिसाल के तौर पे अगर ये वाला बेस पेयर जो है ए है, तो इसके साथ टी लगा देगा। ये अगर टी है तो इसके साथ ए लगा देगा। ये अगर सी है तो इसके साथ जी लगा देगा। तो एक नया स्ट्रैंड इस तरीके से बनता चला जाएगा। अगर ये इन द डायरेक्शन ऑफ रेप्लिकेशन फोक बनेगा, इस डायरेक्शन में बनेगा, तो ये लीडिंग स्ट्रैंड होगा और ये कंटिन्यूअस होगा। लेकिन अगर ये इसकी ऑपोजिट बनेगा, तो फिर ये लैगिंग कहलाएगा, क्योंकि ये लैग करेगा, बार बार डिसकंटिन्यूअस होगा, लैग करेगा। तो इसको हम कहते हैं लैगिंग स्ट्रैंड। और इसमें जो फ्रैगमेंट्स बनते हैं वो मैं कल फिर यहीं से कंटिन्यू करूंगा।
ओके सर, थैंक यू सो मच।
व्हाट इज द फंक्शन ऑफ डीएनए टोपोआइसोमरेज? अभी तो बताया है। टोपोआइसोमरेज जो हैं, टोपोआइसोमरेजेस क्या करते हैं? अ, जो देखो, एक हेलिक्स है। अब इफ आई कैन शो यू व्हाट इज दिस, इमेजिन दैट दिस इज द, नजर आ रहा है आपको? नजर आ रहा है?
यस सर आ रहे हैं।
आ रहा है ये, ओके। ये एक समझें कि हीलिक्स है, सिंगल स्ट्रैंडेड ना। अब जब हीलिकेज इसको यहाँ से ओपन अप करता चला जाएगा, तो यहाँ पे नीचे एक टेंशन डेवलप होती जा रही है और ये सुपर कॉइल्स बनते जा रहे हैं। ये नजर आ रहे हैं आपको, सुपर कॉइल्स, ओके। तो ये जो सुपर कॉइल बना है, इस सुपर कॉइल को रिमूव करने के लिए एक दूसरा एंजाइम आपको चाहिए। उस एंजाइम का नाम है डीएनए टोपोआइसोमरेज। ये टोपोआइसोमरेज इसके ऊपर एक्ट करके ये जो इसकी सुपर कॉइलिंग है ये खत्म कर देगा और हीलिक्स जो है वो स्ट्रैटम हो जाएगा। दिस इज द जॉब ऑफ टोपोआइसोमरेज, ओके। हु आस्क्ड दैट क्वेश्चन बाय द वे? किसने पूछा था ये? मुफलेहा। मुफलेहा, गॉट इट? Could you please repeat last slide? Okay, Farah Feroz, last slide. वैसे लास्ट स्लाइड कल मैं शुरू यहीं से करूंगा, लास्ट स्लाइड से, लेकिन फिर भी आप पूछ लें इसमें क्या पूछना है मुझसे। जी बेटा फरा। फॉस्फोडाइस्टर बॉन्ड की ब्रेक डाउन ऑपोजिट लाइक रेप्लिकेशन, नहीं ब्रेक डाउन नहीं, ये तो सिंथेसिस है। सिंथेसिस ऑपोजिट साइड से होती है, तो जब सिंथेसिस ऑपोजिट साइड से होती है तो हाइड्रोलिसिस भी उन्हीं दो लोकेशंस के बीच में होगी ना, जिन लोकेशंस के बीच में वो फॉर्म हो रहा है। इट इज फ्रॉम 3 प्राइम टू 5 प्राइम। इज दिस डिफिकल्ट? और आसान है या मुश्किल है?